Wonderful discussion, and this set the stage beautifully for our next two graduate students to present their work. You will be hearing after from Erika Beroncal and from Avinash. Both of them will be talking about their studies on organ transplant. We will start with, I believe it's Avinash, you are starting, or is Erika? Erika, you are starting with you presenting in the investigating the relationships between the mitochondria and lung transplant success. Erika, I pass on to you. And after Erika, Avinash Mukala will be presenting on mitochondria transplantation in hepatic ischemia reperfusion lesion, efficacy, mechanism, and transplantation. We are very proud of both of you. Take the stage.
Hi, good afternoon, everyone. My name is Erika Beroncal. I am a master's candidate under the supervision of Dr. Marcelo Sapel and Dr. Anna Andriaza. So I will be talking about investigating the relationship between mitochondria and lung transplant success. So lung transplant is a life-saving procedure that it helps anyone who has end-stage lung disease. However, it only has a median survival rate of 5.8 years, which is the lowest compared to any other solid organ transplant, which is why it's important for us to investigate what we can do to improve these statistics. So one of the things we want to look at is the mitochondria. So first, let's look at the mitochondria in the lungs. The mitochondria, as we all know, is the cellular powerhouse known for its role in ATP production via oxidative phosphorylation. In the lungs, it plays a role in surfactant production under conditions of oxidative stress. And it also plays a role in oxygen consumption for airway responsiveness. What's not on the slide is that the mitochondrial genome is also identified to be a sentinel molecule governing lung cell survival in response to oxidative stress. In a recent collaboration with Anna and Marcelo, we have published a paper in Science Translational Medicine, which is the paper that Marcelo has touched on during the panel discussion, where we looked at 10 degrees preservation that improves lung function during EVLP, which ultimately also protects mitochondrial health in comparison to the 4 degrees preservation, which is currently the gold standard. We have looked at different measures of lung health or lung function, including the peak airway pressure, dynamic lung compliance, and edema or change in lung weight, where we can see that there is a significant improvement for 10 degrees as compared to the 4 degrees. And these improvements have been seen as well in mitochondrial health as measured by CCF-mtDNA, which is released under conditions of mitochondrial stress, mtDNA oxidation, which occurs during when there's high reactive oxygen species. And there's also, we've also measured IL-1 beta and IL-8, which are cytokines measured for inflammation. And all of these have shown that 10 degrees have an improved results and showing that there is a correlation between mitochondrial health and improved lung function, which can be beneficial for lung transplantation. One thing we want to look at, which is which have been mentioned as something that's not yet widely investigated, is the role of mitochondrial haplogroups. So the mitochondria is susceptible to DNA mutations, which is why there are these mitochondrial haplogroups, which are a combination of single nuclear polymorphisms that are an accumulation of sequential maternal mutations. These mutations can happen anywhere in the 37 genes of the mitochondrial DNA or any of the 16.6 kilobase pairs of the mitochondrial DNA, which can then affect an individual oxygen consumption, susceptibility to ROS and mtDNA copy number, and ATP production in their own mitochondria. certain haplogroups have been associated with complex diseases, either at risk or protective from neurodegenerative diseases and cardiovascular diseases. For example, haplogroups F and G have contributed to susceptibility in lung cancer, or haplogroup J, which has low energy efficiency, are less likely to develop chronic renal allograft dysfunction. So in our study, our overall goal is to look at or evaluate the relationship between mitochondrial haplogroups and early lung transplant outcomes. So our aim 1 is to evaluate whether donor mitochondrial haplogroup can be used as a screening tool for risk of development of primary graft dysfunction. As I have said, mitochondrial haplogroups can affect the mitochondria's health. So we hypothesize that donors with haplogroup associated with less efficient mitochondria can give a higher risk for primary graft dysfunction. Aim 2 is to investigate the relevance of mitochondrial haplogroup matching between donor and recipient, preventing primary graft dysfunction. This is under the speculation that when you have different mitochondrial haplogroups, the efficiency of your mitochondria might be different, which could affect the compatibility between the donor and the recipient. Therefore, we hypothesize that recipients and donors with matched mitochondrial haplogroups have lower risk of primary graft dysfunction. In this study, our first quarter, we aim to retrieve tissue samples and extract DNA and RNA. Currently, we are in the process of receiving REB approval. And in the meantime, we are optimizing the protocol for restriction fragment length polymorphism, which is what we will be using for haplogroup determination. Currently, we have seen some successes in the protocol, but it's still under works. In the second quarter, we want to use this protocol for haplogroup determination and sequencing based on the samples that we have collected. And for the third quarter, we will perform mitochondrial DNA analysis, which includes mtDNA oxidation and CCF mtDNA. And finally, we will run the statistical analysis, write-up, and publication. In our study, it will be a retrospective study selecting 100 lung transplant cases, which are divided into 2 groups based on the presence and absence of primary graft dysfunction grade 2 to 3 at day 3 following lung transplantation. Cases and controls will be propensity matched for donor type, either brain death or cardiac death, donor age, and recipient medical diagnoses. We will also be excluding any patients that have HLA mismatch or EVLP cases to avoid confounding variables that could affect the risk of primary graft dysfunction or the mitochondria function. So for the samples, we will be using lung biopsy tissue samples, which we will use for the haplogroup determination and mtDNA oxidation. We will also be collecting plasma prior to reperfusion to measure circulating cell-free mitochondrial DNA and the cytokine levels. The primary outcome of this project is to develop clinically relevant screening tools using mitochondrial haplogroup and function to aid primary graft dysfunction. Currently, we're only using 100 lung transplant samples since it is still a pilot study and we need to create preliminary data first before we can move forward. This project is it's a good way to bridge the gap between the clinical side, the bench scientists and doctors and policymakers and work together to actually move this project forward. And we will be using everyone's expertise to actually which will be behind the success of this project. The surgical team is important for the collection and the banking of human lung biopsy samples. Clinical team will be collecting and recording clinical data, which we will be used for statistical analyses. And the bench scientists will be assessing the mitochondrial biology and quality, focusing on the mitochondrial genome. Meanwhile, the doctors, clinicians, and policymakers are important for us to be able to disseminate the knowledge of what we are able to collect following the conclusion of this project. Patient engagement is an imperative part for this project because we want to provide continuous patient and family education about where the field is going. Being diagnosed with end-stage lung cancer or end-stage lung disease is already very difficult and frustrating, and especially knowing that there is lower success or lung survival, or like shorter lung survival after lung transplant can be frustrating. So we want to ensure that the science is moving forward in somewhere. We're trying to improve these statistics and improve the results of lung transplant, or the successes of lung transplant. So that it will provide ease to the patients. And we want to translate the knowledge that we have and disseminate it about mitochondria and the importance of lung transplantation with the help of our clinicians, doctors, and MITO2i. MITO2i can also provide a platform for patients to voice out their concerns, to help drive the direction of the research, to ensure that we are working with them to provide an output that will benefit both patients and scientists to move the field forward. So I would like to thank my supervisors, Dr. Marcelo Zopel and Dr. Ana Andreasa, and MITO2i for giving us the funding to get this off the ground, and my lab members and the team at University Health Network that are helping me make this project possible. Thank you.